Welcome to our first video on topic 7 of IB biology, nucleic acids. In this video we are looking at DNA structure and DNA replication. So we're going to start off looking at one of the applications here, and this is where you need to be able to understand that Rosalind Franklin and Maurice Wilkins, they investigated the structure of DNA using this technique called X-ray diffraction, and they actually discovered the structure of DNA before Watson and Crick. However, these guys didn't actually publish their work, and what happened was Watson and Crick actually took their understandings and their knowledge, so took Franklin and Watson's Wilkins' understandings and built on it, and then claimed it all as theirs. So you guys know that doing that is actually illegal. It's classed as plagiarism. Okay, so that's why in your IAs you need to ensure that any work that you're using, any previous scientific studies that you're looking at, that you do reference and cite, because as scientists, we need to have those base investigations done so that we can build on it, so we can question research that has already been carried out to see if the correct or the accurate answers have been discovered as such. So that's exactly what Watson and Crick did. There's no issue with what they did, building on the information found out by someone else. However, they didn't reference or acknowledge that Franklin and Wilkins had already done that work using this X-ray diffraction. So here we've got a timeline of work done in the area of genetics. You'll even see that Franklin and Wilkins isn't actually on this list, okay, because Watson and Crick are there and they claimed that as theirs. Okay, but this is a really interesting timeline because it works from Mendel and his work with those pea plants. You can see in 1915 there's Morgan and his fly room. So these are all scientists that we've investigated as part of your IB course. Okay, we're going to be looking at Hershey and Chase now and how they actually proved that DNA was responsible for heredity. Okay. So how was it discovered that DNA is the carrier of genetic material? Well, it's these two guys, Hershey and Chase, who were the scientists who figured this out. Previously, it had been narrowed down to either DNA or proteins being that carrier of the genetic material. Now, how did Hershey and Chase's experiment work? Well, they had some bacteriophage viruses, so viruses that would infect bacteria. They were grown in two different radioactive materials. The first solution was a radioactive phosphorus 32. The second was a radioactive sulfur 35. Now, both of these solutions, so we've got two different types of solution, they had E. coli cells placed in there. Okay, so we've got a mixture with your radioactive solution, your bacteriophage viruses, so viruses that will infect bacteria, and you've got some bacteria, E. coli, in each of these containers. By the nature of a virus functioning, we know that it injects its genetic material into living cells. So these bacteriophages are going to inject their genetic material into the E. coli cells. And we know our structure of our virus, so we know how that all happens. The DNA is in there. It will then inject it through its base plate. Okay, so in this diagram we can actually see what has gone on with these experiments. So we can see on the top line, on the top row, we've got the pink mixture, and this is sulfur 35, just remembering it's not actually pink. The differences in colour is just to show you what the two different solutions as such. So batch 1 being sulfur, batch 2 being the phosphorus. In both cases, you can see we've got our phage attaching to the E. coli, but the difference being in the bottom row, the DNA is radioactive. Okay, so the radioactivity of the phosphorus has attached to the DNA as such, and that gets injected into the pellet, whereas in the sulfur, the actual phage is radioactive. Okay, the protein coat as such, whereas the DNA is not radioactive. Therefore, the final product gives radioactivity in the liquid for the sulfur, whereas not for the phosphorus. So let's just have a look. We go through that here. But why would the radioactivity in the phosphorus 32 adhere to the DNA? So that's what you need to think about. Think about the structure of DNA. Okay, before we get to that actual structure, because that's actually part of your understandings, we need just to be able to build on that, understanding what DNA is, the hierarchy of the structures with DNA. So you've got your DNA strand, parts of that strand are then the genes. You've got the chromosomes, which are the condensed pieces of DNA, that DNA being wrapped around the histones to make those chromosomes, sections of the chromosomes being genes, different genes for the same feature being your alleles, chromosomes in eukaryotic cells found in the nucleus. That's where that genetic material is, whether it's in its DNA raw form or condensed in chromosomes. So just being sure that you understand that background. Also in this unit, we are going to be looking obviously at DNA replication, as we said, so understanding where that falls in the cell cycle is also important. All right, so moving on to your understandings, and this is where we're going to loop back now to that DNA structure. And here we are. Here we've got DNA structure, and this is not new to you. This is just revision, and this is everything I expect you already to know about DNA. Why did the radioactivity in the phosphorus solution adhere to the DNA? We've got our phosphate group in our backbone of our DNA, okay, whereas we've got no sulfur there, no sulfates. Now, a couple of things to remember is we've got our prime ends of our DNA, so our 3 and our 5, remembering what that means. The 3 is the hydroxide end and the 5 is the phosphate end. Obviously that is important when it comes to the direction of replication and other processes. DNA replication, as you know so far. Now there are some terms on here that are new for you, and we're going to look further into that. But we've looked basically at DNA replication. Now we're going to go one step further. So we already know that DNA replication is semi-conservative, meaning that one strand of the parent molecule is found in each of the new daughter strands. We know enzymes split the original strand being that helicase, and then other enzymes create the new strands. How do we know this? Well, this is down to Melson and Stiles' work. They used some E. coli again, grown in a culture of heavy nitrogen, so our N15. This actually was incorporated into the DNA of our E. coli. The E. coli was then removed from the N15 and was then grown in a culture of a light nitrogen. DNA replication then occurred in that lighter culture. What did it show us? Well, some DNA samples were taken periodically and they were analysed for separation. Now, did they sink to the bottom, float on top, or remain in the middle? So the E. coli grown in the N15, that's our red marker here. So that was denser. We've already said the N15 was heavier. The first generation of E. coli is shown in green, so slightly less dense. And then the second generation of E. coli is our blue and our green here. Alrighty, so when we think about DNA replication, we need to remember that DNA is built in a 5 to 3 direction. So remembering 3 is our hydroxide end, 5 is the phosphate end. We are moving from a 5 to 3 direction. So what this means is the enzymes read in a 3 to 5 direction. So it's the opposite. They're reading 3 to 5 and then it's built the opposite because they're doing a base pair, remember. New DNA strand is being built in opposite directions on each strand. So remembering that helicase has unzipped the parent DNA molecule. And then, so what that means, one end would be a 5, so the opposite end from there. One end would be a 3. So what it means is the DNA polymerase that's building the new strands are working in opposite directions on the lagging and the leading strand because those strands start at the opposite value as such. We'll look at that in more depth. My description there was terrible. So first of all, what happens? The entire molecule undergoes the following steps. Gyrase relieves the tensions between the strands. Okay, so gyrase or gyrase, whichever you want to pronounce it. So there's a new enzyme that you've not learned about yet. Helicase untwists the DNA and breaks the hydrogen bonds between the nitrogenous bases to separate the two strands. So we know of helicase already. These are some new ones. Our single strand binding proteins, which we call SSBPs, they bind to the newly separated strands to prevent them from forming new hydrogen bonds. Now, if we didn't have these SSBPs, basically what would happen is helicase would unwind the unzip the DNA and then it would just rezip behind it. So the SSBPs just ensure that it doesn't rezip. All steps after this depends on which strand is then being replicated, the leading strand or the lagging strand, and that is due to the fact that, let me get my diagram, the leading strand is going from the 3 end and the lagging strand is going from the 5 end. So let's look at the leading strand. The DNA polymerase adds complementary nucleotides in a 5 to 3 direction. Did I just say that or did I say it the other way around? This is correct. DNA polymerase 3 adds complementary nucleotides in a 5 to 3 direction to assemble the new complementary strand. Replication is always chasing the replication fork. So what does that mean? The replication fork is where the helicase is unzipping. Okay, so for the leading strand, replication is always working towards the replication fork, so where the helicase is working. And this is called continuous replication. Now our lagging strand, the DNA primase adds an RNA primer in order to tell DNA polymerase 3 where to start. So we've got a slight difference here. We need this primase to add the primer to say this is where you're starting replication polymerase. So then the polymerase adds short sections of nucleotides in a 5 to 3 direction, moving away from the replication fork. So whereas on the leading strand it was moving towards the fork, in the lagging strand it's moving away from the fork. But what it's doing is it's adding sections at a time. And these short segments are called our Okazaki fragments. Okay, so you'll see that on some replication diagrams. You've got that here. Now, DNA polymerase 1 removes the RNA primer and replaces it with the nucleotide that belongs there. Ligase then comes along and attaches the Okazaki fragments together. So when you look at this diagram again now, it makes a bit more sense. You've got these fragments, you've got ligase coming along, attaching them together. We've got DNA primase there, which has told the DNA's polymerase 3 where to start. Whereas, as you can see on the leading strand, it's a bit more simple. Okay, now all this is showing you is replication is very similar for eukaryotic and prokaryotic. However, the difference is that for our prokaryotic, it's the circular DNA, okay, whereas eukaryotic is linear. Okay, an interesting fact, something that you wouldn't have heard of necessarily as such yet, and that is the fact that DNA replication is referred to as being endergonic or endogenic, depending where you are. Energy is required to synthesize the DNA strands, okay? So this is an active process, okay? And that synthesis occurs through condensation polymerization. The nucleotides that are added aren't really nucleotides. Don't you love the way in science how you need to be taught one thing to then make the actual truth make sense as such? So the nucleotides, they're not actually nucleotides. They are deoxynucleoside triphosphate molecules, DNTP. We've got triphosphate there. Very similar to ATP molecules. So when our DNTPs are added by DNA polymerase 3, the phosphate groups are actually removed. And the breaking of this bond provides energy to create the new bonds between the nucleotides. Okay, now, with DNA replication, there can be errors. We've got approximately 4,000 nucleotides being added per second. It's a super fast process. Now, it's important to have large numbers of enzymes and DNTPs available, but some errors can be made. Errors in replication do not always matter, and these can be silent mutations. However, sometimes these errors can be catastrophic. So how is accuracy ensured? Well, we have our complementary base pairing. We have enzymes that can detect and repair any errors. So because base pairing is actually quite simple, it's easy for enzymes to go along and check, but it is a process that's continuously having to be checked for errors because it's happening so quickly. Alrighty, so we move on to one of your application sections now. And also an understanding that links with those applications. So, looking at PCR testing, which you would be hearing so much about at the moment as people are being PCR tested for COVID-19. We know what PCR testing is. We've looked at the process during our standard level course, but just going over it again. PCR stands for polymerase chain reaction. It's a technique used to produce large quantities of DNA very quickly so that they can be studied. DNA is put into favourable conditions such as temperature, enzymes, DNTP, so they've got all of those things available to allow the DNA to replicate very quickly. Now, once that process of PCR was discovered as such or developed, it then allowed the human genome to be mapped. Okay, so why is all this important? Well, it's really looking at that function of DNA, and we know that DNA codes for proteins. That's what you've always been taught. Okay, we know that our genome is a complete set, or a genome is a complete set of DNA. The human genome has over 3 billion base pairs. Most of these base pairs, though, do not code for proteins. They're not genes. So what do they do? 2% of the proteins are coding for genes. 24% are introns. 45% are highly repetitive sequences. 20% are structural DNA, so telomeres, centromeres, things like that. 2% are inactive genes, so maybe genes that have historically been used but no longer are required. And there's 7 to 8% that we have no idea why they are there. Okay, so the human genome was sequenced using Sanger's method. Single-stranded copies of DNA are placed into 4 different test tubes, each with plenty of DNTPs, enzymes and primers. So you've already looked at Sanger's method before. We're going into a little bit more depth now. In each tube, one of the DNTPs has been replaced with DDNTP, so deoxynucleoside triphosphate. And that's just showing you the difference between those there. Okay, so deoxynucleotide has the hydroxide group there, whereas deoxynucleotide just has a hydrogen. Once the DDNTP is attached, chemical bonding cannot occur and replication stops. However, anywhere that DDNP is added, replication stops and the strand is cut short. Strands of different lengths are loaded into gel electrophoresis. Bands enable you to determine the sequence. So this is how we look at those gene sequences. So if we have a look at this, we've got our DNA to be sequenced, then our template strand, we've got DDATP, we've got DDGTP, DDCTP, DDTTP. Okay, so each of these are stopping at specific bases. Okay, so then in tube 1, it chopped it into those sections, tube 2, chopped it into those sections, tube 3, into those, tube 4, into that. And then we can see how that looks on the template there. So highly repetitive sequences, STRs as they are known, are short tandem repeats. Everyone has them in some location, but differ in the number of repeats. It gives different sizes of DNA and can be run through gel electrophoresis to determine the size of DNA. And it's this that's used then for your DNA profiling to solve crimes, paternity, etc. Okay, so again, before we move on, just have that understanding of the cell cycle, where our DNA replication occurs at that S phase of the interphase. Okay, we are now looking at a couple of understanding and a skill. Looking at nucleosomes. Right, in electron micrographs, nucleosomes look like beads on a string. So you can see over here, here's our nucleosomes, and they look like beads on a string. This association is to help organize our DNA, regulate gene expression, and simplify mitosis-meiosis. If we didn't have nucleosomes for our DNA to condense around, then replication wouldn't be able to occur because the DNA would be all intertwined together. When we say chromatin condenses into chromosomes during the prophase, what we're actually referring to is nucleosomes supercoiling to form dense, organised chromosomes. and we're going to watch that now.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
That's the end of 7.1. I didn't put the last slide in. I look forward to seeing you in our next video.